Good day and welcome to One-on-One Maths Private Tutors. Today we are looking at O-Level and IGCSE Biology for pure biology and combined science. We continue with plant nutrition. The topic is leaves. The shape of the leaf makes it ideally adapted to carry out its functions of absorbing light for photosynthesis and allowing gases to diffuse into and out of the leaf tissues. Leaves have a large surface area to absorb light rays. They have a thin shape so gases can diffuse in and out easily over short distances. They have many chloroplasts to absorb light, which provides energy for the reactions that take place in photosynthesis. They have a network of veins to support the leaf's surface and to carry water and ions to the leaf cells and to take sucrose and amino acids away from the leaf to other parts of the plant for use and storage. Inside a leaf, to find out how a leaf works, we can look at the structure of a typical leaf. So the aim of this topic is to find out what the structure or what type of cells make up the leaf from the top surface to the bottom surface. When we cut across a leaf, what structures do we find inside the leaf? The external structure of the leaf consists of a midrib, the upper epidermis, and the lower epidermis. Now, if we cut a section through the leaf from top to bottom, we have a structured layer of the leaf. The first layer is what we call the upper epidermis, and it consists of the cuticle, which is a waterproof layer that also cuts down the water loss by evaporation. So it's a waxy waterproof layer that prevents water loss by evaporation. And below it is a row of cells that are thin and transparent to allow light to enter into the palisade mesophyll layer underneath it. Then we have the palisade mesophyll tissue, or a layer of palisade mesophyll cells. These are column-shaped cells that are tightly packed with chloroplasts to absorb more light, maximizing photosynthesis. Then below the palisade mesophyll tissue, we have the spongy mesophyll tissue. These contain internal air spaces that increase the surface area to volume ratio for the diffusion of gases, mainly carbon dioxide. Then we have the lower epidermis, which contains guard cells and stomata. So we can see the guard cells. These two are called guard cells that open and close to allow the movement of carbon dioxide into the cell. These guard cells absorb water to open and lose water to close the stomata to allow carbon dioxide to diffuse in and oxygen to diffuse out. So these are responsible for gaseous exchange. Carbon dioxide that is needed by the leaf for photosynthesis moves in, and oxygen that is produced as a byproduct of photosynthesis moves out. The guard cells also help in preventing water loss. The stomata is the opening between the guard cells. This is where gas exchange takes place. It opens during the day and closes during the night. Evaporation of water also takes place from there. In most plants, stomata are mostly found on the underside of the leaf. Then we have the vascular bundle. Here, this contains the xylem, so the top part is the xylem, and phloem, which is the bottom part. The xylem is responsible for bringing water and mineral ions from the roots to the leaf, and the phloem is responsible for carrying sucrose and amino acids around the plant. So the vascular bundle is part of the network of veins that we find on the leaf, distributing water and mineral ions to cells in the leaf and taking away sucrose and amino acids produced by the leaf to other parts of the plant. So vascular bundle contains xylem and phloem to transport substances to and from the leaf. The upper part is the xylem, which transport water and mineral ions into the leaf from mesophyll cells to use in photosynthesis. So that is the structure of the leaf from top to bottom. So we can see the cells that contains most chlorophyll are the palisade, the sponge mesophyll, and the guard cells. So this is the structure of the guard cells. The inner walls are thick and inelastic. The outer walls are thin and elastic. So during daytime, guard cells become turgid by osmosis of water into the guard cells. Water enters inside the cell and the guard cells become turgid. The turgor pressure is exerted on the thin walls of guard cells. So these elastic and thin walls expand and stretch out. The thicker in walls are pulled apart. Then the stomata opens. At night, guard cells become flaccid because water is moving out. When they lose water, they become flaccid, closing the stoma and reducing water loss. So that is the operation of the guard cells to open and close the stomata. Now we look at adaptations of the leaf structure for photosynthesis. Here we are looking at how the shape and the structure of the leaf makes it suitable for photosynthesis. The leaf has a large surface area. This helps because it increases the surface area for the diffusion of carbon dioxide and absorption of light for photosynthesis. The leaf is thin. The distance from the top to the bottom of the leaf is very small. This helps in allowing carbon dioxide to diffuse a short distance to palisade mesophyll cells. The availability of chlorophyll in the leaves helps in absorbing sunlight energy so that photosynthesis can take place. The network of veins on the leaf, which make up the vascular bundle, allows the transport of water to the cells of the leaf and carbohydrates in the form of sucrose from the leaf to be transported to all other cells. Water is needed for photosynthesis. Carbohydrates or sucrose as well as amino acids are a product of photosynthesis. The availability of stomata allows carbon dioxide to diffuse into the leaf and oxygen to diffuse out. The upper epidermis is thin and transparent. This allows more light to reach palisade cells. The cuticle is thin and made of wax. This protects the leaf from water loss without blocking sunlight. The availability of the palisade cell layer at the top of the leaf maximizes absorption of light, as it will hit chloroplasts in the cells directly. The spongy layer helps in allowing carbon dioxide to diffuse through the leaf in order to reach palisade cells. The vascular bundles with thick cell walls help to support the stem and the leaf. Now we move to mineral requirements. Plants need more light, carbon dioxide, and water for healthy growth. They also need mineral ions, also called plant nutrients. The nutrients are absorbed from the soil in small quantities as ions by active transport in the roots. Plant nutrients. Nutrients are needed for healthy growth of plants. They are also used for a variety of purposes in plants. If these nutrients are lacking in the soil, then the plants do not grow well and show certain symptoms known as deficiency symptoms. Plants need nitrate ions to make amino acids, which are used to make proteins. As proteins are required for growth, plants deficient in nitrates show poor growth. Magnesium ions are absorbed by plants and used to make chlorophyll. Leaves of plants deficient in chlorophyll look yellow, a condition known as chlorosis. Plants also need phosphate ions for making compounds such as DNA and for respiration. If the soil does not contain enough nutrients, farmers and growers may add more as fertilizers to replace the missing soil nutrients. So the diagram shows a leaf that has magnesium deficiency. Since magnesium is used for chlorophyll, the green pigment required for photosynthesis. Gas exchange in plants. All living organisms, including plants, carry out respiration all the time. Plants are no different from other organisms. To carry out respiration, they need a supply of oxygen, and they produce carbon dioxide as a waste. At night, plants exchange these gases with their surroundings. During the day, plants carry out photosynthesis as well as respiration. In bright light, when there is a high rate of photosynthesis, some of the oxygen produced in their chloroplasts is used by mitochondria for aerobic respiration. The rest of the oxygen is not required, so it diffuses out of the plant. The carbon dioxide produced by mitochondria in respiration is used by chloroplasts for photosynthesis. However, this is not enough, so carbon dioxide diffuses in from the surroundings. Gas exchange in aquatic or water plants is investigated with hydrogen carbonate indicator solution. This solution contains hydrogen carbonate to provide carbon dioxide and 2 pH indicators. Carbon dioxide is an acidic gas. When it dissolves in water, it forms carbonic acid, which is a weak acid. Hydrogen carbonate indicator solution is prepared by bubbling atmospheric air through it. This gives the indicator a red color. Let's look at this practical to investigate photosynthesis and respiration in plants. Set up 3 test tubes as shown in this diagram. One of the test tubes is covered in aluminum foil. Add hydrogen carbonate indicator solution to each test tube. Cut 2 pieces of pondweed 10 cm in length and place one in each of the test tubes 1 and 2. Put test tube 1 in darkness by covering it with a foil. So this is the one that is in darkness, but there is the same length of pondweed in there. Leave the test tube near a light for 2 to 3 hours. If carbon dioxide is added to the water by the plant, the hydrogen carbonate indicator solution will turn from red to yellow. If carbon dioxide is taken up from the water by the plant, the hydrogen carbonate indicator solution will turn from red to purple. So in test tube 1, the indicator turned yellow because carbon dioxide was released by the pondweed during respiration. So in 1 here, there is no photosynthesis taking place since there is no light into the test tube. So the only process that is happening here is respiration. which releases carbon dioxide. So if carbon dioxide is added to the water, red to yellow, so it will turn yellow. In test tube 2, the indicator turned purple because carbon dioxide was taken up by the pondweed for photosynthesis. Now, the purpose of test tube 3 was to act as a control. So test tube 3, there is no pondweed there. It's just our indicator solution in water. The indicator solution only responds to changes in carbon dioxide. The color changes have nothing to do with oxygen released by the plant in the light or absorbed by plant in the dark. When oxygen dissolves in water, it does not change the pH. If you wanted to monitor the changes in oxygen concentration, you would have to use an oxygen probe and a data logger. If pondweed is kept in a test tube of hydrogen carbonate indicator solution at low light intensity, the color of the solution usually remains red, as the rates of photosynthesis and respiration are the same. With that, we come to the end of the section. Thank you.